Friends, welcome to Biology Made Easy. Today in this video, we'll discuss the remaining part of the nucleus. In the previous video, we have started the nucleus where we have discussed the number of nucleus per cell, the size, shape, and also coming to the structure, we have discussed that the nucleus consists of four parts. That is, the outermost part is nuclear membrane, and the details of nuclear membrane and the pore structure we have discussed in the previous video. Then today we will discuss the second part is the nucleoplasm, which is also known as Karyosome, or it is also known as nuclear shaft. And the third one is that is chromatin, and the fourth is the nucleolus. These are the four parts of the nucleus. And in the previous video we have discussed this one, where we have discussed that the nucleus is surrounded by a double membrane structure, and there are pores are there. So this is the nucleus, and the outermost membrane of the nucleus, several ribosomes are attached, while the inner nuclear membrane is smooth and the outer membrane is continuous with the endoplasmic reticulum. And what is the structure of the nuclear pores, that we have discussed in the previous video. Now let us start the second one, that is nucleoplasm or Karyosome. Now inside this nuclear membrane, there is a matrix-like structure, there is a viscous fluid-like structure, and that fluid-like structure is a viscous fluid, and this viscous fluid is known as nucleoplasm. That means just like cytoplasm is present within the cell membrane, but the same fluid when present inside the nucleus is known as nucleoplasm, or it is known as nuclear shaft, or known as Karyosome. Now, what are the different substances present within this Karyosome? The different substances they are present. They are usually the nucleosides, then different types of enzymes, and the prominent enzymes are that is DNA polymerase, RNA polymerase, and also that is nucleoside phosphatases. These are the different types of enzymes present, and along with that the histones, non-histone proteins are also present here. The histone proteins, then the non-histone proteins. So all these things are present within this. This is known as the Karyosome. Now in this Karyosome, the outermost part of this Karyosome is also known as nuclear matrix, and just below this nuclear membrane there is a network of some fibrous-like structure. There is a network of fibril-like structures like this, just below the inner nuclear membrane. So this structure, which is present in the outermost part of the nucleoplasm, just inner to this nuclear membrane, this is known as nuclear lamina. And it consists of different types of lamins. So lamin proteins. So lamins A, B, C. These are the different types of proteins that are present. Now, what is the function of this nuclear lamina? You should be able to distinguish between this is the nuclear lamina, while the different proteins which control the exchange of materials between in the nuclear pore, that is known as nucleoplasmin. but this is nuclear lamina. Now, what is the important function of this nuclear lamina? Usually, it maintains the shape of the nucleus. So, it usually maintains shape of nucleus. Then it gives a mechanical strength. Then within this nucleoplasm, the third one, chromatin, is present. So it provides a scaffold to the chromatin network. So it provides scaffold for chromatin. And another point is the chromatin, the telomeres of the chromatin, they are attached to this nuclear lamina. The telomeres of chromatin are attached. And another thing is they form some of the they form the components of this nuclear pore. That means they give rise to components of nuclear pore. So these are the important functions of this nuclear lamina, and main function is it provides the shape and mechanical strength to the entire nucleus. So that is about this the second one, that is nucleoplasm or karyosome leaf. Then let us discuss the third one, that is chromatin. Now within this nucleoplasm, there are some thread-like structures present in a reticulate manner. Likewise, some thread-like structures are present in a reticulate manner, like this. This is known as chromatin. So this is known as this is chromatin. Now this chromatin, it is studied only at the interface stage, and it will give some basic stains like acetocarmine, or if let us give any other stain, that is maybe basic fusin, then the chromatin will get colored. Now this chromatin of interface stage becomes condensed to form chromosomes during S phase of cell division. Now here some important points we'll discuss, so listen very carefully. Now each chromatin has only one DNA. This is very important. Each chromatin has only one DNA. But we know that each chromosome has two DNA. So what happens? During S phase, this chromatin, it doubles. So that in S phase, each chromosome has two DNA molecules. That means, suppose you take the case of human beings. In case of human beings, 2n is called 46 chromosomes are there. That means when they are present in this interface stage, in interface, there are 46 chromosomes, means 46 chromatins are there, and means 46 DNA are there. That means in human cell, at interface 46 chromosome means 46 chromatins, 46 DNA molecules are there. But in S phase, when we will move to S phase during the cell division, the number of chromosomes will be 46 chromosomes. But here there will be duplication of DNA, there will be synthesis of DNA. So there will be 92 chromatins and 92 DNA molecules. That means the number of chromosomes remains same, but the amount of DNA becomes doubled. Now, what is the amount of DNA That also we can measure. Now when we are telling that 2n is equal to 46, this indicates the number of chromosomes. When we tell that 2n is equal to 24 in case of potato or tomato, that means that is the number of chromosomes. But if you take the weight of that chromosomes in haploid set, then that is known as C value. C value. And C means chromatin. C means chromatin. That means what is C value? So C value means the amount of DNA in haploid set of chromosomes. In a haploid set of chromosomes, what is the amount of DNA? Now in a haploid set, the number of chromosomes is n. But what is the weight? This is number, but what is the weight? That weight is known as C value. This is the difference. Now let us give one example. Suppose we will take 10 numbers of potato, 10 brinjals and 10 onions. So in each case the number is 10. But when we will weigh 10 potatoes, the weight will be 1. 10 brinjals, the weight will be different. 10 onions, the weight will be different. That means the number is same, but weight becomes different depending upon the size. So similarly, suppose in case of suppose potato, in case of potato 2n is equal to 24, in case of tomato also 2n is equal to 24, in case of brinjal also 2n is equal to 24. That means in each case the number is same, but the size of chromosomes are different. That is why the C value will be different. So here C means the chromatin value, the amount of DNA that is present, and that is measured in picograms. The C value is measured in picograms. So what is the amount of DNA in interphase or at G1 phase? At G1 phase, the amount of C value is 2C. Suppose it is 2C because the number of chromosomes are also 2n number of chromosomes. But in S phase, there will be synthesis of DNA. So during S phase, it will be 4C because there is synthesis of DNA. So during again G2 phase, it is also 4C. Then prophase, metaphase, again it will be 4C. But after anaphase, there will be again separation, then it will return back to 2C. So this is the difference between the number of chromosomes and the chromatin value. The number is counted in form of digits, while the C value is the amount which is taken in the form of picograms. So this C value differs from cell to cell. It depends upon the amount of DNA that is present. So I think now this aspect is very clear. That is, suppose you take the case of human beings where there are 46 chromosomes, means at the interphase 46 chromatins, means 46 DNA. But when S phase, the number of chromosomes will remain constant, but the amount of DNA will be doubled. That means C value will be doubled. This is the important aspect of this chromatin. Then another important aspect is when this chromatin is stained with by either acetocarmine or basic fusin, it is found that the entire chromatin is not uniformly stained. There are some light parts, while some are very darkly stained parts like this. Some light and some dark parts are there like this. So those which are a lightly stained part, that is known as euchromatin. Euchromatin, and those which are darkly stained, that is known as heterochromatin. And the euchromatin which is a lightly stained, that is genetically active. Genetically active, that means it can undergo transcription, it can give rise to mRNA, it can undergo crossing over, all those things because it is genetically active. But heterochromatin, where there is more amount of DNA, because there is more coiling, this is genetically inactive. It is genetically inactive. So this is the difference between this euchromatin and heterochromatin depending on the staining behavior. But at certain places, this coiling of the DNA is so much, sometimes it is so much that it may give the false appearance of a nucleolus. As if somebody will think that this is nucleolus, but this is not a true nucleolus, this is false nucleolus. So when there is more staining, this is known as a false nucleolus, or we can call it as karyosome, or we can call it as chromocenter. That means the false nucleolus or karyosome is actually not a nucleolus, but there is more coiling of the DNA, so it gives the appearance of a nucleolus. So this is about euchromatin and heterochromatin. Euchromatin is genetically active, while heterochromatin is genetically inactive, and it undergoes late replication and late crossing over. So now the chromatin part is over. Now the next part is the nucleolus. So inside the nucleus, there is another structure which is known as nucleolus. The nucleolus is, it was discovered by Fontana. Nucleus was discovered by Fontana. So discovered by Fontana 1731. And this nucleolus is usually, it is membraneless. That means it is not surrounded by any membrane. Then how it is distinguished from the nucleoplasm? It is distinguished by, because it is darkly stained. It is darkly stained. That is why it can be distinguished. That means here suppose this is one, this is the structure. Now this one is nucleolus. So this nucleolus is membraneless, but it is darkly stained so that it can be distinguished from the remaining part. Now how many nucleoli are present in a particular nucleus? Usually the number varies from 1 to 4 per nucleus. But in case of Xenopus, it is found that there are 1600 nucleoli. So what will be the number of nucleoli? That again depends upon the activity of the nucleus, activity of the cell. Because the nucleolus gives rise to ribosomes, and ribosomes are responsible for protein synthesis. That means the cells which are more active for protein synthesis, there will be more number of nucleoli. That is why in cancer cells there are more number of nucleoli. In case of cancer cells, more nucleoli are there because there is more. And this nucleoli, it consists of DNA. Chemically it consists of DNA, RNA and protein. And these three are they stabilized together due to presence of calcium ions. It gives a calcium ions, stabilize them. And another important aspect is, when we will discuss chromosomes, you will know that there are certain chromosomes where there is secondary constriction, or there is a NOR part. NOR means a nucleolar organizing region in certain cells, and they are usually known as short chromosomes. Usually this nucleolus is attached to that short chromosome. So Suppose this is one chromatin, and here there is one secondary constriction. This is the NOR part. The NOR part, this is also known as the secondary constriction. Secondary constriction, and in this NOR part, there is specifically rDNA, and this rDNA it transcribes rRNA, means ribosomal RNA. So this ribosomal RNA, it combines with the proteins that comes from the cytoplasm, ribosomal proteins. So the ribosomal proteins comes from the cytoplasm and the rRNA. They combine together and they give rise to ribosomes. Because the nucleolus is the site of synthesis of ribosomes. So for that, for synthesis of ribosomes, it transcribes rRNA. The rDNA transcribes rRNA and the ribosomal protein that comes from the cytoplasm, and they combine together, and the rRNA combines with the proteins, and it gives rise to ribosomes. It gives rise to ribosomes, and the two separate units are formed separately. The bigger unit is 60S, the smaller is 40S. So the nucleolus is the factory of synthesis of ribosomes, and the NOR part is attached to that part. So this is the nucleolus, that is attached to this. Now if we take the ultrastructure of this nucleolus, again it has 4 parts. The nucleolus, though it is membraneless, but it looks like this. Don't think that these are the membranes. And the nucleolus, this is the diagram of nucleolus. It consists of 4 parts. So what are the 4 parts? Now this one matrix-like structure is there. This is known as number 1, pars amorpha. That means this is simple matrix. Then here you will find certain granule-like structures like this. Some granule-like structures are there. Granule-like structures. These are known as number 2, pars granulosa. Pars granulosa, and these are the precursors of the ribosomes. You can write ribosomal precursors. Then there are certain fibril-like structures like this. So these are known as pars fibrosa. These are known as number 3, pars fibrosa, and these are nothing, they are the RNA fibrils. And these are actually not membranes. These are also, they are part of the DNA. So this is known as the number 4, is known as pars chromosoma, or also it is known as perinuclear chromatin, perinucleolar chromatin. So this is the ultrastructure of the nucleolus. It is completely membraneless but darkly stained, so that we can distinguish it from the nucleoplasm. So this is the structure of the nucleus, where there are 4 parts: nuclear membrane, nucleoplasm, chromatin, and nucleolus. Now what is the important function of the nucleus? That is very common. So when we discuss the function, nucleus contains the genetic material. So when we will discuss the functions, now it is the storehouse of genetic material. storehouse of genetic material. That means it contains the DNA, the genes, and that controls all the activities. Then it controls all activities, of the cell by giving rise to RNAs. RNA is this mRNA, tRNA, RNA. The DNA produces all this, and they give rise to proteins, enzymes. So through that transcription and translation, all the physiological activities are carried out. Then another function is meticulously divide. To transmit characters from generation to generation. So during cell division, that is during mitosis, the nucleus divides meticulously because the chromosomes have to be equally distributed to the two daughter cells, and that is the reason why all the cells of a living organism are same, because it is of the meticulous cell division. And again, this is nucleolus, is the site of synthesis of ribosomes. So site of synthesis of ribosomes. So these are some of the important functions of the nucleus. Now I hope now a very clear understanding about the structure of the nucleus, nucleolus, and the different aspects of it along with the functions. So thank you very much for watching the video. Wish you a very bright future. God bless you.